Microcystis is commonly given the nickname of blue-green algae, even though it is not an algae, but a bacteria. It is a gram-negative bacteria that only lives in freshwater environments. It belongs to the phylum of cyanobacteria, meaning it is a photosynthetic organism. The species we are going to be most interested in is Microcystis aeruginosa, simply because it is the most toxic one. These tiny gas vacuoles are a unique feature. The bacteria can inject gas into them to reach the surface and get all the oxygen and all the light it needs for its growth, while also outcompeting other water organisms. This is how it is able to form impressive blooms like this one at Lake Erie 2011. There are specific conditions in which those blooms will be promoted, including when you have nutrient-rich water, for instance, because of agricultural runoff or dysfunctional wastewater treatment. Another condition could be relatively high temperature, so usually in summer. The reason why this bacteria is toxic to humans and animals is because it can release an endotoxin called microcystin. Because it's an endotoxin, it is released only when the cell dies and lyses. Microcystin is a really short peptide, only 7 amino acids. It has a cyclic structure, and there are actually over 60 different types of microcystins. The different microcystins have different levels of toxicity, but they are all hepatotoxins, which means that they target the liver. Symptoms can go from dizziness, headaches, nausea, vomiting, to death from liver failure in the more severe cases. Although it should be noted that this mostly happens to wild animals and cattle because they have the highest levels of exposure. There isn't really a single infective dose for microcystins because the effects are gradual and they are varied. But the EPA and the WHO do recommend that there should be no more than 1 microgram of microcystin per liter of water for consumption. Once released by the bacteria, the toxin can remain in water with a half-life of 1 week. Or it can go hide in sediments and dry scum and stay there for months. Then people, but especially animals, can be exposed directly in the environment by ingestion of the contaminated water or even following consumption of shellfish, in which the toxin can be biomagnified. This is also a risk for drinking water treatment plants that draw their water from such contaminated surface waters because of the contamination by the toxin itself, but also because of modification in taste, odor, turbidity that are due to the presence of the algae. Different detection methods exist to target the algae or its toxin. Culture-based detection methods aim at distinguishing between the different species of Microcystis based on morphological differences. Then you have to provide the right conditions for Microcystis bacteria to grow. This includes a moist culture media, minerals, and of course, light. However, this is not that reliable. And what we actually want to know is whether the bacteria is able to produce a toxin or not. To do this, we can perform a PCR of the whole cell and target genes that are involved in the production of the toxin. But again, this doesn't tell us whether the toxin is present in the water, because the toxin is released only when the cell dies. So we actually need detection methods for the toxin itself. Liquid chromatography and mass spectrometry is the most precise method because it is able to distinguish between the different types of microcystins that have different levels of toxicity. However, it is quite expensive and long. Another simpler method is the colorimetric protein phosphatase inhibition assays, in which you track the inhibition of the phosphatase enzymes by microcystins. Commercial kits also exist for the ELISA assay of microcystin. These last two methods are cheap, easy to operate, and quantitative, but they do not allow you to distinguish between the different types of microcystins, and the risk of cross-reactivity is really high. There are several categories of treatment methods. You can try to treat the issue at the root by removing the algae from the surface water. Methods that have proven efficient include promoting mixing or using sonication that will disrupt the gas vacuoles of the algae and make it sink. Nevertheless, you should be really careful because this can lead to a sudden release of cyanotoxin following the death of the cyanobacteria. Most commonly used drinking water treatment processes are actually quite efficient at removing the cyanotoxin, the most efficient ones being activated carbon and ozonation. Chlorination can also be used, with a CT value that is similar to that of Giardia. But chlorination efficiency is highly dependent on pH. Finally, microcystin is highly resistant to UV treatment. However, in the case of a bloom, the drinking water treatment plant should be really careful in choosing its water intake and pre-treating the water. If too many algae enter the plant during a bloom, they may clog filters and reduce the efficiency of oxidation processes because of increase in organic load. Fortunately, strategies like bank filtration or selection of water intake depth exist to avoid this type of situation.